Okay, we'll try and see what at least you'll know your lab grades going into this. Very often that final thing, unless you really bombed one test, you can't change the overall grade that much. Okay, so the first thing we did is, I guess, male reproductive. So to realize that if the testes in the fetus started out in your abdomen, they then descended through the inguinal canal, and what it was, the labia majora on the females becomes closed on a male to be a scrotal sac. The scrotal sac has inside it has a dartus muscle which can tighten up and somewhat raise the testes a little bit if the weather's too cold. Because it came from the abdomen, it's surrounded in this tunica vaginalis, which is a serosal membrane. And then the testes, outermost layer is this tunica albuginea, which is connective tissue. All the blood vessels and nerves that had innervated the testes continue through that inguinal canal, so it maintains its connection, along with this thing called the vas deferens, which leads out to the urethra. And the cremasteric muscle wraps around them. Can also raise the testes when it's too cold. The reason why the testes are in the scrotum is so that it can be 3 degrees cooler than body temperature for sperm production. And the whole thing with the wrapped cremasteric muscle and the vas deferens and the blood vessels and nerves and lymph, that all is called together a spermatic cord as it goes through the inguinal ligament. Within the testes itself, the connective tissue divides it up into these different lobules. They're filled with seminiferous tubules. Under FSH, that's where sperm is produced. The germ cells are called spermatogonium. When they start to differentiate, they divide, one of them will differentiate into a primary spermatocyte. That's destined to undergo meiosis. After the first meiotic division, because there's going to be 2 of them, it's now called a secondary spermatocyte. And after the second division, it's now called a spermatid. And the spermatid will develop a tail and become spermatozoa. Cells inside the seminiferous tubules which guide that development from spermatogonium all the way to spermatozoa are called nursing cells or sustentacular cells. They respond to FSH. And the process of creating that spermatozoa is known as spermatogenesis. with meiosis, different from mitosis in that prophase 1, the homologous pairs become entwined in what's called synapsis. They synapse with each other, and we create a tetrad because there's two paired chromatids that wrap around each other in a synapse. We now have 4 strands, or a tetrad. That's what gets pulled apart. The homologous pairs get pulled apart in the first division. So it's known as a reduction division. Within the seminiferous tubules, there are certain cells, as I tried to point out on the microscope, that are not part of seminiferous tubules. Those are called interstitial cells or Leydig cells. They respond to LH and produce androgens, like testosterone. Once the sperm is made in the seminiferous tubules, it moves to the superior posterior portion of the testes, known as the retestes, and they exit efferent ducts into the epididymis, where they might remain for a couple of months and undergo capacitation. From the epididymis, from the head to the tail, it then goes up the vas deferens. The vas deferens goes behind the bladder, where it enlarges as an ampulla of the vas deferens. The ampulla of the vas deferens meets with the seminal vesicles, in the ejaculatory duct, and will then empty into the prostatic urethra. From the prostatic urethra to the membranous urethra, and then the spongy urethra. The erectile tissue of the penis is the corpus spongiosum on the ventral surface, and 2 corpus cavernosum on the dorsal surface.
Isn't there a word that was similar to spermatogenesis? Like,
Yeah.
different,
Spermatic, yeah. Spermiogenesis was when it starts developing a tail. It's like a subset of spermatogenesis. But as long as you know spermatogenesis, I don't think I used that other word. So at the base of the corpus spongiosum is the bulb, and at the tip of the penis, that's called the glans penis, the swelling at the tip. And there's a muscle that wraps around it, the bulb, called the bulbospongiosus muscle. And there's also one that wraps around the crust of the corpora cavernosum, called the ischiocavernosus muscle. Those 2 muscles become active with ejaculation.
What was the second one? The second muscle?
Ischiocavernosus. So the arousal phase is parasympathetic. It allows the cavernosum to become engorged so that the male maintains an erection. And then the emission phase is where that seminal fluid and fluid and sperm from the vas deferens move into the ejaculatory duct. But the final stage, and that's usually sympathetic, and the final stage is conscious, it's the cortex, contraction of those other two muscles, bulbo and ischial.
So, because there's three phases, the parasympathetic, does it apply like the initial phase?
Yeah.
So from ejaculation to emission is all sympathetic.
Well, the emission, they say, is sympathetic, and then they say the ejaculation is cortical, because those are skeletal muscles. The other accessory organs, I mentioned seminal vesicles. There's also, seminal vesicles usually has clotting factors in them and some fructose for energy. Then there's the prostate gland, which has a milky secretion, a little bit acidic, but it helps activate the sperm, and also has factors that will unclot the sperm when it reaches the cervix. And then the last gland was the bulbourethral gland, which buffers the acids that might have been in the, from urine that might be in the urethra. Bless you. So the sperm are necessary to provide, not the sperm, the semen from those accessory organs are necessary to provide nutrients for the spermatozoa and help propel it along the reproductive tract. And they also buffer and activate the sperm. The nursing cells will help in the production of, well, they'll coordinate spermatogenesis. And they help form like a blood-testis barrier. They tend to have this androgen binding protein that holds testosterone within that region. And you find them from the wall of the seminiferous tubule all the way towards the lumen. The more androgens we release, it's under negative feedback. So it will decrease the levels of gonadotrophic releasing hormone, and we'll wind up having less FSH and LH release. But testosterone is responsible for the male secondary sex characteristics, its growth, deeper voice, muscle development, bone development, increased sex drive. That's all a result of testosterone. Going to the females, all their oogonium are gone by the time of birth. They all become primary oocytes while they're embryos. The 2 million primary oocytes by the time they're born. And they're stuck in prophase 1 of meiosis. And that's the way they'll stay until puberty. So once puberty occurs and FSH is released, the follicles that surround those primary oocytes are stimulated by follicle-stimulating hormone. And they thicken and become granulosa cells, which release estrogen. And that's the female secondary sex characteristics developed from estrogen. So once a girl reaches puberty, she's already lost most of the oocytes she was born with. There's still about 400,000 of them left, and you only release about 400 eggs in a lifetime, so several of these eggs never will wind up being released. No oogonium in an adult female. When the egg is released, when the as each cycle, when one follicle tends to really enlarge, it becomes a secondary follicle, then becomes a Graafian follicle. The meiosis 1 is completed in the Graafian follicle. So we're left with a polar body that's released and one secondary oocyte. That at this point, estrogen levels are very high and LH is released. LH causes the Graafian follicle to rupture and release the secondary oocyte. Typically, that secondary oocyte will not undergo its second meiotic division unless a sperm has first contacted it and bound the receptor on the membrane. That stimulates the second meiosis. So the gametes for the male is the sperm and the ova for the female. The gonads are the testes for the male and the ovaries for the female. So as the eggs develop in the ovaries, in the cortex of the ovaries. And again, the ovaries are within the pelvic cavity. They're not retroperitoneal, like most of the rest of the female reproductive tract is. And when the egg is fertilized, as it becomes a zygote, that will occur within the oviduct. And then the embryo, which is a blastula, at the time it reaches the uterus, must implant itself into the uteral wall. So the uterus serves for mechanical protection, but also provides nutritional support for the developing embryo. The thing I pointed out in the lab when it was number 6 on that side wall near the sinks was I was looking for the cervix. The cervix bulges into the vagina. On either side you have the fornix of the vagina, but number 6 was pointing to the cervix. It was part of the uterus. Uterus has a fundus on top, a body in the middle, and a cervix below. The uterine tube, or the oviduct, has fimbria, which is what I was more or less looking for with those red tips, as part of the infundibulum. Then we have this little larger portion, the ampulla, and then the isthmus. Know the different cycles, the way we looked at them. There's a uterine cycle, which looks at when estrogen is raised, it's a proliferative phase. Then there's ovulation, and then we go into a secretory phase. because luteinizing hormone, because progesterone is largely present then, and the uterine wall will become more lush. So during the proliferative phase, it follows menses, because that's what comes next after the secretory phase is the menses. And the proliferative phase restores that stratum functionalis, the functional zone of the endometrium. The stratum basalis always remains there. But each cycle, it's the stratum functionalis that gets thicker, thicker, thicker. It becomes secretory and is then sloughed off. If we're talking about ovarian cycles, we're looking at a follicular stage in which follicle-stimulating hormone is stimulating the follicles to develop and create one secondary oocyte and one grafting follicle. Then we have ovulation, and then the luteal phase, when luteinizing hormone causes ovulation and the establishment of a corpus luteum. Women will release, I said, about 400 to 500 eggs in a lifetime, usually by the age of 45, 55. Their uterine cycle stops. It's known as menopause. And they'll no longer be releasing eggs. And the good news is they'll no longer be menses once a month. They'll just be hot flashes. The external genitalia consist of the labia majora, the fatty folds with hair. Underneath that is the labia minora, the fatty folds without hair, and that opens up into a vestibule. The anterior portion of the vestibule is the clitoris. Behind that is the urethral opening, and behind that is the vaginal opening. Menstrual cycle is the sloughing off, I said that already, of the endometrial layer of the uterus. Vagina just serves as a conduit for removal of that. It also serves as a conduit for receiving the penis. And will protect the baby during pregnancy. Forms that lower portion of the birth canal. Now to the lymph. Look at the main functions of the lymph. We talked about it in the beginning. It returns the interstitial fluid back into circulation. And the other main, bless you, the other main function is it's providing immunity. So the different, when we talk about lymph tissue, it will either be a lymph tissue which isn't encapsulated. That's mainly your mucus-associated lymph tissue. Doesn't have a connective tissue capsule around it. It's just within the mucosa and the submucosa. Peyer's patches is an example of mole. We have in our throat, we have tonsils, pharyngeal tonsils, lingual, palatine tonsils. Again, there's no capsule around that. but they're clusters of lymphocytes and presenting cells. They have little germinal centers where the excited lymphocytes might be cloning. Then we have lymph organs. Lymph organs are encapsulated. The two primary ones are the bone marrow, which is where B cells mature, and the thymus, which is where T cells mature. And the other lymph organs, considered secondary, that's the lymph nodes and the spleen, which is where these naive B and T cells become active effector cells. That can occur in these organs or can occur in the lymph tissues. Spleen, in addition to a spot where you can activate B and T cells, also is for the removal of old red blood cells. Look at the different things that were part of the innate or non-specific defenses. Nothing to do so much with lymph, but they're all present even when we're born. We don't have to acquire immunity to them by first noting an antigen. Inflammation, interferons, complement, natural killer cells, macrophages, even our epithelial cells form a barrier that's part of an innate non-specific defense. Fever is a non-specific defense. The only T cell that's kind of non-specific is those natural killer cells. But cytotoxic T cells, helper T cells, and B cells are all part of the specific immune system, the acquired or adaptive immune system. Yeah?
Isn't the neutrophils, eosinophils, those are part of the non-specific?
Not specific. Eosinophils, neutrophils, they'll attack anything. They don't need a specific antigen. The others are very specific. And so once they encounter something, they also develop memory. Eosinophils and neutrophils never develop memory to anything. So phagocytes, they start as, the large ones, the macrophages, they start as monocytes in the blood. Usually when they leave the blood, they're leaving because of there's been an inflammation, and by chemotaxis, they move through the walls. The slide called it emigration, or I called it also diaphoresis, the way they squeeze through. They then will adhere to some toxins that might be there or foreign substances, and they then phagocytize them, making them a macrophage. They phagocytize. They put little cytoplasmic extensions around foreign material and create a vesicle that they pull inside. These vesicles then merge with lysosomes so they can be destroyed. All T cells require activation by antigen-presenting cells. That provides their co-stimulation. CD4 cells bind MHC2s to become effective helper T cells. And they will then secrete interleukins. If you can remember they secrete interleukins, I think that's good enough, because I don't think I asked you, was it interleukin 2 or interleukin 1 or anything? Just interleukins. Interleukins 2 stimulate other lymphocytes to undergo rapid cloning. But just to know they secrete interleukins. And every T cell matures within the thymus. If they can't recognize MHCs, or if their T cell receptors are responding to self-antigens, they're eliminated or destroyed. So CD8 cells will bind MHC1s. And when they're activated, they will destroy cells that present with abnormal antigens, specific abnormal antigen on the MHC1s. So they're effective against cells that have been attacked by viruses, cells that have become cancerous, or cells that have been transplanted into the body.
Professor?
Yeah.
So the CD8 cells, they bind to MHC2 or MHC1?
MHC1, CD8, CD4s are binding to 1.
And it destroys cells that present specific.
What?
Abnormal.
Yeah. In other words, they're not every every cytotoxic T cell's gonna attack a given microbe. It's only if they specifically are responding to that antigen they have.
So what binds to MHC2?
To help the CD4s.
CD4s? Okay.
So the B cells, they perform what's called a humoral response. They see things in liquid. They're not necessarily looking on MHCs for things. They're not at all. They don't see things on MHCs. They can attack things that are, or they'll be sensitized by antigens floating in fluids, interstitial fluids, lymph, or blood. And once they're sensitized, they need helper T cells to activate them. Activation of a B cell results in cloning of plasma cells that produce massive amounts of antibodies. IgG antibodies are the most common, and they fight most infections. IgMs are often the first produced, but we don't develop as many of them as we do IgGs. IgAs are secreted out the body, mainly in mother's milk, as an example. And IgEs are associated with allergies. They typically bind mast cells. Their constant portion sits on mast cells. So if their variable portion sees some antigen, it will activate the mast cells to release histamines.
And
Basophils? Yeah, maybe, I don't know if I didn't hear that they sit on basophils. Basophils also release histamine. For the most, no, I cracked myself up. That's funny then. You probably won't find it funny, but what was it? Oh yeah, helper T cells. You know, they're going to assist both cytotoxic T cells into cloning and plasma cells into cloning. So they have roles for both cell-mediated response, which is the cytotoxic, and humoral immune response, which is the antibodies. T cells that are not specific are those natural killer T cells. They provide immunological surveillance and look for any deviation on the MHCs. They don't care about the antigen that's in there, they just look for abnormal MHCs. Leukocytes or lymphocytes sometimes also release, especially these cytotoxic ones, they release interferons. But so do other cells. So a cell that's attacked with a virus will often release interferons. It's too late for the cell that was infected. It doesn't protect them releasing interferons, but it serves as a paracrine messenger, and it helps other cells ward off the virus.
Was that the lymphocyte or
Interferons.
interference?
They're released from either cells attacked by viruses, or sometimes they're just released by lymphocytes. They're chemical messengers, cytokines. Not specific to the lymph though, those are lymphokines. The two different types of antigens we discussed were endogenous antigens, which means it comes from within a cell itself. Those will be always displayed on MHC1s. Exogenous antigens, which tend to be larger in many cases, they're put on these MHC2s. Foreign cells or that have been transplanted, cancerous cells or virus cells tend to display endogenous antigens on MHC2s. Excuse me, endogenous antigens on the MHC1s. Cancerous cells, viral infected cells, or transplanted cells. Those are endogenous, they make those up themselves. But if there's toxins or pollen or things floating in the blood that macrophages gobble up, they'll tend to put those things, tend to be larger peptides. They put those on MHC2s. Know the basic characteristics of this adaptive or acquired immune system. Number one, you know it has specificity. We developed, this is from Jenner's work in 1798, there's specificity that the cells will attack only a specific pathogen. And they have memory. If they've encountered that pathogen before, they'll have a swift response the second time. And an acquired immune response can be induced. Sometimes it's induced naturally. Sometimes it's induced artificially, as in a vaccine. Naturally, as you just encounter it with someone else that's sick. And the immune system recognizes self and leaves self alone. Also, it's very versatile. It can recognize billions of antigens. Presenting cells can either be dendritic cells, most common, macrophages, and sometimes B cells. They all serve as antigen-presenting cells to the T cells.
Or B cells, is that the last one?
T cells.
Oh, yes, sir. So, B cells can become APCs. Do they actually phagocytize the
They don't phagocytize it, but the receptor on the top, which is an IgD, when it becomes bound, it moves in by endocytosis.
okay.
Similar to phagocytizing, but it just moves in. It's bound, it comes in. It forms an endosome rather than a phagosome.
mix it up.
It still goes for lysosome, just the same, yeah. Yeah, phagocytosis, I look at as a specialized endocytosis. It's just just undergoes endocytosis. Almost done. So we have innate immunity, and we discussed what those things were. You create tears, we have acid in the stomach, we have epithelial barriers. Any one of the white blood cells other than lymphocytes are all part of our innate immunity. You asked me a question in the book, the way that some diseases fish have, we don't get. That's got to be innate, because right from the start we don't get those diseases. Then there could be induced or active immunity. If it's going to be induced, I didn't put a slide up on this, did I? Well, active immunity can occur naturally. That's just as a child. I know we didn't have vaccines when I was young. If someone had mumps, go and play with him. We'd rather the kid got mumps and became sick than he was an adult, or a woman who's pregnant and gets mumps for the first time and has a miscarriage. So you kind of let your kids play with other kids that were sick so that you'd get those illnesses, and then you have immunity. That's naturally acquired active immunity. Now we induce it by having vaccines. That's induced, it's an active immunity versus passive immunity. If a baby is taking mother's milk, that's passive immunity. She's getting antibodies, the baby's getting antibodies directly. But if you give them a vaccine, they're actively promoting their own immunity. Hadn't talked about those before,
Then there's an artificial passive.
An artificial passive? Well, I'm saying artificial passive. Artificial passive. I don't know about artificial, but there's passive where you take.
There's an active passive would be vaccine, but
Active, that's not passive. That's induced.
Induced,
Active,
like active,
yeah.
so artificial active.
I don't know. Yeah, I guess. Sorry, but we call it induced
There's
active.
no passive artificial.
No. But passive immunity is just taking mother's milk in.
It's based on the environment.
Yeah. Well, naturally acquired immunity comes from the environment. So we'll know what an antibody looks like. They're called immunoglobulins. They have 2 heavy chains, 2 light chains. They have a constant region, they have a variable region, and they activate complement and participate in opsonization. Makes it easier for phagocytosis to take place. So they'll see things that might be floating particles within liquid and attack it. They can't see anything that's inside the cell, though. If something's going wrong inside the cell, antibodies can't do anything about that. But cytotoxic T cells can, because if something is going wrong inside the cell, there's going to be abnormal proteins displayed on the MHC1s. So cytotoxic T cells take care of things going wrong inside the cell.
Antibodies are just markers?
What?
Antibodies are just markers for what's abnormal?
Well, for one thing, they're markers. They can serve as markers. They can immobilize the microbes. They can cause them to clump. And by activating complement, they cause them to burst too. And they're markers. They opsonize them, make it easier to swallow, and also by chemotaxis kind of pull macrophages into the area. Yeah, certainly any foreign antigen, and sometimes they overdo it because all of a sudden they're mounting this full-blown response against pollen, and you don't want that. I mean, the pollen's not going to kill you, but it doesn't know. So know the difference. IgG is the most common. They fight infections. IgD is what's found on the surface of every B cell. IgM are often the first responders. IgE are attached to mast cells and are used for allergies. So if you hear the word classical and alternative pathways, or Properdine is the alternative pathways, Properdine, those are pathways that we activate complement. The classical one is through antibodies. And an antigen has this epitope, which is considered an antigenic portion of it. That alone will not elicit an immune response. It has to be big enough to elicit an immune response. So some haptens are antigenic, but unless they attach to a larger protein or substance, they won't elicit an immune response. Poison ivy works like that. Penicillin allergies work like that. In and of themselves, their molecules are too small, but they can bind to larger things. Those are called haptens. Know the classic signs of inflammation: the heat, redness, swelling, and pain. I didn't talk as much about this, but HIV, that's considered a retrovirus. By retro, it means it's RNA, but when it goes into the cell, its RNA becomes converted to DNA. That's the retro of it. And then it controls the cell. HIV tends to, it's a retrovirus that tends to be taken in by helper T cells. So they destroy the functioning of helper T cells. Helper T cells can't be activated because they're too busy making viral parts. So we saw that the first time you encounter an antigen, you're likely, or a pathogen, you're likely to get sick by it, but on a second encounter, there'll be a quick, swift rise in your antibody titer, and often symptoms will never be realized. The effects of aging: you get more autoantibodies, your thymus shrinks. You get a weakened immune response. You respond less to vaccinations. And then looking at the B cells, how they're, look at the stages that occur from a B cell being naive until it releases antibodies and binds some antigen and causes opsonization. Be able to put that in order. From the moment the B cell, naive B cell, first binds the antigen on its IgD surface. That was with the last slide. And that would be the end of it.
How many questions are there?
That had like